Hello, my name is Jeremiah Campbell. I'm a graduate student at the University of Texas at Arlington, and today I will be discussing the role of endothelin-1 and L-Arginine in blunted sensory nerve-mediated cutaneous dilation in Black women. Cardiovascular disease is a leading cause of morbidity and mortality worldwide, and in the Black population, they have higher prevalences of hypertensive and cardiovascular diseases relative to other populations. While the reason for this elevated CVD prevalence is multifactorial, it is believed that impaired micro and microvascular function is likely a contributing factor. Our lab has previously reported impaired microvascular function as indexed by impaired sensory nerve mediated vasodilation in college aged Black men and women. This response was restored following the pharmacological inhibition of NADPA oxidase and xanthine oxidase in Black men only, but no effect in Black women. This data these data suggest divergent mechanisms of impaired microvascular function between Black men and Black women. Two possible mechanism pathways are heightened endothelin 1 activity and reduced L-arginine bioavailability. Therefore, we tested the hypothesis that ET-1 overactivity and arginine insufficiency contribute to blunted sensory nerve-mediated cutaneous dilation in Black women. We investigated 7 young Black women aged 21. Subjects were studied after an overnight fast and was abstained from alcohol, caffeine, and strenuous activity for at least 24 hours. Four intradermal microdialysis membranes were placed in a dorsal forearm using a 25 gauge needle. Red blood cell flux was assessed at each site using laser Doppler flowmetry housed inside a local heating unit. Breakthrough blood pressure was taken through a electrical spirometer. Pharmacological agents that was used is our lactated ringers as our control, BQ123 as the ET receptor type A antagonist, BQ788 as the ET receptor type B antagonist, L-arginine, and L-name. L-name was used to inhibit nitric oxide synthase and thus determine the nitric oxide contribution. The exogenous donors, SMP, was used to elicit a maximum vasodilation at each site. The effect of BQ123, BQ788, and L-arginine on sensory nerve mediated vasodilation during the initial peak was assessed during a standard 39 degrees Celsius local heating protocol. Site-specific maximum continuous blood flow was determined via combined SMP infusion and increasing the temperature to 43 degrees Celsius. Data then was represented as cutaneous vascular conductance response during the initial dilation and is reported as percent of site-specific maximum. So looking at figure one, this is the schematic representation of what's happening with the intradermal microdialysis. So we have the membrane, which is inserted underneath the skin. We have pharmacological agents, is which is infused through the skin, and then laser Doppler flow probe measuring the red blood cell flux. The local heaters are then turned to 39 degrees to increase vasodilation and then set to 43 degrees Celsius to look at maximum dilation. In figure two, we are looking at the protocol, the schematic protocol for the entire experiment. So after 60 minutes of trauma resolution, each site is infused with their accordingly drug for 30 minutes for a wash-in. After that, the temperature and the skin temperature is clamped to 33 degrees Celsius for 10 minutes. Then the temperature is raised to 39 degrees Celsius for 30 minutes. Then we add the L-name into NOS inhibition for 30 minutes at 39 degrees Celsius. And the last steps we do is we infuse SMP for 2 to 3 minutes and increase the temperature to 43 degrees Celsius to achieve maximum dilation. So, and lastly, in figure three is showing you what's happening typically in a local heating response. So what we're focusing on is more of this initial peak for our study here. The results include we have our CVC max on our y-axis, and then we have our white women, which we got from results previously in our lab, and our Black women, looking at the control for the Black women, comparing it to the control for the white women, and looking at BQ123, BQ788, and L-arginine. As you can see from this data here is that when we added the BQ18123, BQ788, and the L-Arginine, it did not return the function, the max vasodilation function to what we've seen in the white women. Therefore, contrary to our hypothesis, the previously observed blunted sensory nerve-mediated vasodilation during a local heating protocol is not related to ET-1 overactivity or L-arginine insufficiency. Future research should be aimed at characterizing the impairments in vascular function and vasodilatory capacity in Black women is warranted. Thank you for your time and have a good day.